So here's a possible explanation for why you can have a suppressor which does not interfere with the wild-type function of the locus that the suppressor acts on when mutant. As you know from your biochemistry courses, biochemical pathways that lead to amino acids and related compounds are quite complex and branched often. So here's a hypothetical pathway leading to methionine, and let's say that the MET1 locus encodes an enzyme that's necessary for this particular step, so taking one precursor and passing and converting it to another, which then goes on later in the pathway to become methionine. If this function is removed in the MET1 minus mutant, then this pathway is blocked at this location and cannot complete to make methionine, resulting in methionine oxytrophy. Now, the suppressor could be an enzyme, for example, on the related branched pathway producing some compound that we'll call X that's closely related to methionine. The loss of the suppressor enzyme, or the loss of this enzyme, the mutant form now, that we've designated as the suppressor allele, might block this pathway at this location. So now we have a block there. What can happen in some situations is because this pathway is now blocked, this component can increase in concentration in the cell. And it can increase in concentration enough, build up enough in the cell because of this blockage, that this enzyme, whatever this is, if we put in enzymes at each of the steps, as this compound increases in concentration, it might well be that it can stand in for this compound. And now this enzyme might be able to use this as a substrate to convert to methionine, simply because once the concentration increases to a sufficient level, that it's enough to drive this reaction forward through this enzyme. So as a result, having a block here with this loss of this enzyme can act to suppress the loss of this enzyme, simply because we can now bypass the defective step in the methionine biosynthesis pathway. So this sort of pathway suppressor type of situation will, when recombination is allowed, what you can see is, well, what would happen if we had MET1 plus with the suppressor? So now we've got a functional MET1 plus enzyme here, so this pathway can now complete just fine. And we have the suppressor allele, so this non-essential pathway is blocked at this location. You can see in this situation we're going to get methionine made without any trouble. First off, the first pathway, the standard methionine pathway, is available and completely functional if we have MET1+. And this suppressor allele is not blocking the function of anything that's essential. And if anything, it's going to spill over some of this precursor into the methionine biosynthesis pathway. So in this particular setup with a sort of pathway suppressor, this genotype will be prototrophic because it will be able to make its own methionine. This is also what's called a non-heterodimer suppressor, which we'll contrast in a moment with a heterodimer suppressor. So you don't yet know what a heterodimer suppressor is, but this is also known as a non-heterodimer suppressor, and why that's the case will become obvious in the next clip. Now that we've examined pathway or non-heterodimer suppressors, it's time to examine heterodimer suppressors, and what that means will become obvious in a moment. So again, we're dealing with this genotype, and now this is an alternate situation to what we were discussing before with the pathway suppressor. In the case of a pathway suppressor, we decided that this genotype would be prototrophic because the suppressor doesn't interfere with the wild-type function of the MET1 locus, the wild-type allele at the MET1 locus. In this case, however, with a heterodimer suppressor, this genotype is actually oxytrophic, and we'll explain why, how it is that it can be that way. Again, we've got some biosynthesis pathway that's leading to methionine, and we have different enzymes that are catalyzing different steps as we take different compounds through the pathway to methionine. Now, these different enzymes that do the different steps are here just represented with these circles. But let's just take this one and say blow it up a bit and have a look at it. It may well be that this enzyme is actually composed of two interlocking proteins that form a dimer. A dimer just means a pair or two proteins acting together in this context. And this is a heterodimer if in fact these proteins come from different loci. So if you have two proteins that come from different loci and they bind together to form a dimer, we call that a heterodimer. Hetero just meaning different, dimer meaning in this context two proteins gathering together to do a function. And it may well be that one or any number of these enzymes in this pathway requires two different polypeptides from two different loci to form together and to make the enzymatic function. So now what we could say is, okay, perhaps one of these loci is what we've identified as the MET1 locus. So if that's MET1 plus, we might imagine that the MET1 minus mutation, the original mutation event that converted MET1 plus to MET1 minus, may have altered the shape of this protein. Now, there's a lot of different ways to make mutations in proteins that range all the way from deleting the locus to eliminating expression, altering a splice junction, all that sort of thing. This is a special case where we're talking about an amino acid substitution that alters the physical shape or tertiary structure of that polypeptide. What could happen in this situation is the binding pocket for its interaction with the other polypeptide might be altered. So in this context, there's actually not an ability for these two polypeptides to associate with one another anymore. So as a result, this no longer forms a complex, and we've now lost an enzymatic function en route to methionine. Now, there's one way to fix this that we haven't talked about before. One way to fix this and to restore binding would be to have a similar or complementary mutation that occurs in this other polypeptide to alter its binding shape, such that when we had the phenotypic reversion event, we may have been able to put these two polypeptides back together again. So now we have the MET1 minus allele and the suppressor allele, which are now working together. So if this restores function, this would act as a suppressor of the original MET1 minus mutant. The thing is now when we have recombination, you can see that the wild-type allele of the suppressor, if now we call this the suppressor plus, the wild-type allele of the suppressor plus is not going to be able to interact with the MET1 minus. So if we looked at this particular genotype here and drew out what the proteins looked like, we would have the wild-type allele of MET1, and then we would have the suppressing allele, the allele of the suppressor that acts to suppress the MET1 minus mutant. And you can see these two things, because now this has been mutated to match up with the MET1 minus allele. These two don't bind, don't form a heterodimer. A homodimer would be two peptides from the same locus that are associating with each other. So these don't form a heterodimer, and as a result, we don't have enzymatic function. As a result, this would have an auxotrophic phenotype. So in the case of a heterodimer suppressor, we have two auxotrophic phenotypes and two prototrophic phenotypes. And if you look back at the previous videos, you'll see that in a non-heterodimer or a pathway type suppressor, this genotype has a prototrophic phenotype. So the way you tell the difference between this specialized case of a suppressor where it's acting as a heterodimer is to determine the phenotype of this genotype. This is the recombinant that has the suppressing allele in the presence of the wild type locus of the original mutant. If this has an auxotrophic phenotype, that tells you that you're dealing with a heterodimer suppressor and that the wild type of the original locus and the suppressing allele cannot form a dimer together to function. And as a result, we have two auxotrophic genotypes in the progeny. So remember, these are the progeny that are coming from the original revertant times wild type cross. So that's what we're using to form these progeny. And if the revertant was originally a heterodimer suppressor, then these are the recombinant genotypes that you would get, and this specific genotype, these are the genotypes you would get. Of the recombinants, this one with the wild type of the original locus and the suppressing allele won't work because they can't physically associate with one another, and as a result we have two auxotrophic genotypes. So the key to distinguishing whether or not you're dealing with a heterodimer or a non-heterodimer suppressor is the phenotype of this genotype. If it's a heterodimer suppressor, this will be auxotrophic. If it's a non-heterodimer suppressor, this will be prototrophic. In the case of a prototrophic phenotype for this genotype with the non-heterodimer situation, then you will have only one auxotrophic genotype. This one will always be auxotrophic, regardless as to whether or not it's a heterodimer or not. But in the case of a heterodimer suppressor, this genotype will also be auxotrophic. So that's the key to distinguishing between those two types.